Next topic is crassulaceous acid metabolism under the chapter photosynthesis. As name suggests, crassulation, that is concerned with the family Crassulaceae. Crassulation means concerned with the family Crassulaceae. Crassulacean plants, for example Crassula. They have thick fleshy green leaves. You can also see in Bryophyllum plant. Bryophyllum leaves are also thick, fleshy and green. And a speciality is concerned with these plants, that is their stomata open at night. Their stomata open at night and close during day. Stomata open at night and close during day. Such stomata are known as scotoactive stomata. So crassulacean plant, they have scotoactive stomata, that is they open at night and close during day. Second physiological speciality related with them, that is these plants synthesize enormous amount of organic acid at night. These plants synthesize enormous amount of organic acid at night, they synthesize enormous amount of organic acid at night, and that acid is particularly malic acid. For the formation of malic acid, they use atmospheric carbon dioxide. That is, they use atmospheric carbon dioxide at night. Another special feature, these plants fix at atmospheric carbon dioxide at night and form malic acid. These plants fix atmospheric carbon dioxide at night and synthesize malic acid. This is anabolic part of them. This is also termed as dark acidification. This is also called dark acidification. That is synthesis of organic acid at night. That is dark acidification, and this is anabolic part of these plant. Next day during daytime, they consume that acid. That is, the formed malic acid is consumed during day, that is formation takes place at night and use or consumption takes place during day. So during day deacidification, that is catabolic part. This is catabolic part. Formation of acid and consumption of acid, that is acid metabolism. Specifically seen in the members of family Crassulaceae, that's what is called crassulaceous acid metabolism. That's why it is known as crassulaceous acid metabolism. Let's see how they form in these plants. The mesophyll cells, they are mesophyll cells. When stomata are open, they have PEP like C4 plant, 3C compound, PEP, that is 3C compound, phosphoenol pyruvate, and PEP has great affinity towards carbon dioxide. This PEP traps atmospheric carbon dioxide at night and forms oxaloacetic acid, 4C compound, and it is reduced to malic acid in same way as we have studied in CAM plants. This malic acid is formed as a reduction of oxaloacetic acid. This malic acid which is formed at night, it is stored in the vacuole. And this malic acid is basically concerned with the opening of stomata at night, because malic acid dissociates into malate plus H plus ion. Malate plus H plus ion. Malate plus H plus ion. These H ions are replaced by potassium. So potassium malate is formed. And potassium malate governs stomatal opening. At night. During daytime, this malic acid is consumed. This malic acid is released from the vacuole. Stomata are closed now, and the malic acid is released from the vacuole and undergoes decarboxylation. And the carbon dioxide which is released, it is utilized by the chloroplast during daytime for the photosynthesis process, especially in Calvin cycle. Malic acid formation at night. Malic acid is formed at night, or malate is formed at night. This undergoes decarboxylation during day, and this carbon dioxide is released. Stomata are closed, so carbon dioxide can't be taken by the plant from air, and this is the only source during daytime. So during daytime, malate undergoes decarboxylation, releases carbon dioxide, and this carbon dioxide is used by the chloroplast. To perform Calvin cycle. So this is the only source for the carbon dioxide during daytime to operate Calvin cycle. Normal photosynthesis takes place during day. Daytime when light is available. But for that, carbon dioxide is available from internal reactions. That is, malate get converted into pyruvate and carbon dioxide is released. Malate is converted into pyruvate and carbon dioxide is released. And this is the reaction for the source of carbon dioxide. The special feature, stomata are closed, so neither oxygen is taken in nor carbon dioxide is taken in by the plant during daytime. And oxygen also produced in the photolysis of water, it is utilized by the plant tissue for the respiratory activity. So there is no exchange of gases during daytime from external environment. There is no exchange of gases during daytime. All gases are available from the internal reaction. During daytime, CO2 from internal conversion, oxygen due to photolysis of water is available for the plant tissue. This is crassulation acid metabolism. 3 specific feature. Number 1, thick fleshy green leaves. Second, stomata close during day, open at night, that is the scotoactive stomata. And third speciality, synthesis of organic acid during night and use of acid during the daytime. These 3 main features are there. So this is one more speciality. This is for the specific physiological adaptation to prevent excess loss of water during daytime. This is all about crassulation acid metabolism. Next activity, that is photorespiration. Photorespiration. Photorespiration is seen in C3 plants. First, it is seen in C3 plants. Photo indicates light, involvement of light. Respiration, that is oxidation. Respiration, that is oxidation. Photo, respiration, 2 terms are there. Photo pertaining to light, respiration, that is oxidation. The special type of oxidation which is observed during the daytime when light is available, and only by the C3 plants, but conditionally. This is photorespiration, that is a special type of oxidation which is performed in presence of light. The condition required, that is carbon dioxide is low. Low means below compensation point. Low means below compensation point. This carbon dioxide should be below compensation point. Compensation point means at which photosynthesis is equal to respiration. This is called compensation point. That is the least concentration of carbon dioxide, or that critical concentration of carbon dioxide at which the rate of photosynthesis is equal to the respiration. That is compensation point with respect to the carbon dioxide. If carbon dioxide is not there up to the compensation point, in that case atmosphere is already rich in oxygen. Atmosphere is already rich in oxygen, and green plants, that is C3 plants, enzyme is there, that is rubisco, ribulose, bisphosphate, carboxylase and oxygenase. The dual behavior of this enzyme, dual nature of this enzyme, that is involved in carboxylation as well as oxidation. And in both the cases, the substrate is RUBP. The substrate is RUBP. This RUBP. RUBP, if CO2 is sufficient, that is at compensation point or above the compensation point, then RUBP prefers carboxylation. If carbon dioxide is below compensation point, in that case RUBP undergoes oxidation. This RUBP undergoes oxidation by using oxygen. In both the cases, I have already mentioned this enzyme supports Rubisco. At the time of carboxylation, its activity is carboxylase. At the time of oxidation, it performs oxygenase activity. So, conclusion, that is conditional oxidation, spatial and conditional oxidation of RUBP in presence of light when there is a low carbon dioxide and high oxygen concentration. Better to say carbon dioxide is below compensation point. In that case, RUBP oxidizes, and this entire process is known as photorespiration. This activity is called photorespiration. In photorespiration process, chief metabolite glycolic acid is formed, or glycolate. This is chief metabolite. Glycolic acid or glycolate. Chief metabolite, a C2 compound. Glycolic acid or glycolate is chief metabolite of this process. See this one. As a result of oxidation of RUBP, first of all, 2-phosphoglycolate is generated. It loses its phosphate to water and converted into glycolate. So, glycolic acid or glycolate C2 compound, it is chief metabolite. Why metabolite? Because it is formed in this process as well as consumed. Glycolic acid is formed in this process as well as consumed or undergoes catabolism also. Formation and catabolism. That's why photorespiration is also known as glycolate metabolism or C2 cycle. Photorespiration is also called glycolate metabolism or C2 cycle. In this process, there is a role of 3 cell organelles. That is, the process accomplishes in 3 cell organelle, and according to their order of participation, number 1 chloroplast, this activity, that is formation of glycolic acid, takes place in the chloroplast. Then second one is peroxisome, a microbody. It is also known as uracosome. It is also called uracosome. And third structure is mitochondria. These 3 cell organelles are involved. Question pre-medical, that is number 1 chloroplast, number 2 peroxisome, and number 3 mitochondria. And due to this particular activity, that is photorespiration, there is a sharp decline in the rate of photosynthesis. Photosynthesis reduced to more than half, 1/3 or less, and ATPs are also consumed in the photorespiratory activity. ATPs are also used up in the photorespiratory activity. So, in brief, let's see the involvement of 3 cell organelles. Number 1, the process starts with the chloroplast. First of all, oxidation of RUBP results in the formation of glycolate. This glycolate is transferred to peroxisome. Where its oxidation takes place and a amino acid is generated, that is glycine. Glycolate undergoes oxidation, formation of glyoxylate, and by the union of a amino acid, glycine is formed. Glycine is formed. So one important thing, during photorespiration, two amino acids are also produced. Two amino acids are also produced. These are glycine. These are glycine. And the second one is serine. Second one is serine. So glycine and serine, these are also produced during the photorespiration activity. Glycine production is associated with the peroxisome. It is associated with the peroxisome. Then glycine migrates or transferred to the mitochondria, where its decarboxylation and deamination takes place, results in the formation of serine as well as its oxidation, oxidative deamination plus decarboxylation. Oxidative, that is, this is the oxidation step, and this is decarboxylation, then removal of ammonia, that is deamination. So we can say oxidative decarboxylation plus deamination results in the formation of serine. Serine is the second amino acid produced during photorespiration. First one is glycine, second is serine. Serine is again transferred back to the peroxisome and it undergoes the association of ammonia to form hydroxypyruvate, followed by its oxidation takes place. This is oxidation. Oxidizer is NAD and glycerate, a 3C carbon compound, is generated. Glycerate is produced. Glycerate is transferred to chloroplast. Glycerate is transferred to chloroplast, where it uses the ATP of light phase. And converted into 3-phosphoglyceric acid. Actually when RUBP oxidizes, it produces two compounds, first one and this is second one. So this is also obtained by the glycolate also, as well as directly from the RUBP, So this is the photorespiration. The key points, better to see the key point. Number one, process takes place in 3 cell organelles, that is chloroplast, peroxisome, mitochondria. Second, chief metabolite glycolate. Third, glycine and serine, these two are also produced during photorespiratory activity. Then in this process, oxidizer is NAD. In this process, oxidizer is NAD. And ATPs of light phases are also consumed. So this is photorespiration, and due to photorespiration, the process of photosynthesis is reduced, and with the consumption of ATP also. So conditional process, during daytime if photorespiration takes place, you can see the atmosphere of carbon dioxide around the leaf. If carbon dioxide is there around the leaf during daytime, especially during daytime, then definitely the process of photorespiration is taking place. Why? Because carbon dioxide is generally used during daytime in the photosynthetic activities. But here the photosynthesis process is almost stopped and the process of photorespiration is going on, which produces carbon dioxide in mitochondria, especially in mitochondria. That's all for the photorespiration. That's all for the photorespiration. Finally, come to the factors affecting photosynthesis. Factors affecting the rate of photosynthesis. Photosynthesis is actually constructive process, that is, plant synthesize carbohydrates. It is concerned with the yield of plant, especially in crop plants. And there are a number of external and internal factors are there which affects the photosynthetic activities. Before starting the factor, a law is applied in the photosynthesis, that is Blackman's law of limiting factor. Blackman's law of limiting factor, and this law is based on law of minimum. Law of minimum. This was given by Liebig. This is given by the Liebig. The Blackman's law of limiting factor is a modification of law of minimum. In this process, or according to this law, suppose a process is affected by number of factors. For example, photosynthesis, affected by light, factor 1, affected by temperature, factor 2, and affected by the carbon dioxide concentration. These three factors suppose affects the process. In that case, according to this law, see this statement. When a process depends on number of factors, rate is limited by the pace of slowest factor. The rate is limited by the slowest factor. I mean to say, factor light, factor temperature, and the third one is carbon dioxide. Both are affecting this process at same time. In all those conditions, check the factor which is minimum or least available to the plant. In that case, if that particular factor is increased, the rate of photosynthesis is also increased. If it is decreased, rate of photosynthesis also decline. So that factor is the slowest factor, and it enhances rate till it crosses its limit. That is, now it is not a minimum, till then it enhances the rate. I mean, when we supply, this is photosynthesis, rate of photosynthesis, this chart indicates rate of photosynthesis, this is carbon dioxide concentration. Carbon dioxide concentration. Suppose this is in least in those conditions. When the rate becomes constant, that is at low light intensity. Beyond this, carbon dioxide does not increase the rate, because in those conditions, the requirement of carbon dioxide is over by supplying it. Suppose there is a medium intensity of light, then more carbon dioxide is needed. But at this condition, what amount of carbon dioxide is required by the plant? If it is given, the rate after that rate will not increase. That is, now it is not a limiting. Rate enhances till that factor should be minimum. That factor enhances the rate till it is minimum. This is law of limiting factor. Same with, at this highlight intensity, up to this limit carbon dioxide is required. Up to this limit carbon dioxide is required. Beyond this, rate will not increase. Rate will not increase. In medium intensity of light, this much amount of carbon dioxide is needed. In low light intensity, this much amount of carbon dioxide is needed. So the law says the factor which is in minimum, that is slowest factor, it influences the rate, or the rate can be enhanced or enhanced or increased by the supplying of that factor. And this chart already indicates this is carbon dioxide concentration, that is, if low intensity is there, then this much amount of carbon dioxide is needed. Beyond this limit, carbon dioxide will not be limiting factor. If medium intensity of light, this much amount of carbon dioxide is required. After that, rate will not increase. At high light intensity, this much amount of carbon dioxide is required. Beyond this, rate will not increase. I mean, there is a certain amount of carbon dioxide in those conditions, till that, only up to that limit, rate can be controlled. After that, that factor will not be limiting factor. The factor which controls the rate, is known as limiting factor. The factor which controls the rate in those conditions is known as limiting factor, or relative factor in relative minimum. We can say it factor in relative minimum, factor in relative minimum, or it is also known as most significant factor. Most significant factor. This is Blackman's law of limiting factor. Let's start with the external factor. External factor, first one is light, and source of light is sun. Sunlight reaches to the earth's surface by radiation process. So solar energy is also called radiant energy. And light is studied under three headlines. Number 1, intensity. Number 2, quality of light. And number 3, duration of light. Let's start with the intensity of light. For C3 plant and C4 plant, this is optimum light intensity. That is, C3 plant requires 1000 to 1200 foot candle light for optimal photosynthesis. And C4 plants require more intensity light. I have already described C3 plants, they prefer to live in temperate climatic condition, whereas C4 plants are fit for the tropical climatic conditions. So if intensity of light is limiting, then only it increases the rate. If intensity of light is a limiting factor, then you supply the highlight intensity, the rate will increase. But at very high light intensity, it causes solarization. Very high light intensity, that is beyond bearable intensity. In that case, photooxidation of photosynthetic pigments and chloroplast. Chloroplast damages due to highlight intensity, and instead of enhance, the process is completely stopped. Solarization is harmful activity. Then come to the quality of light. photosynthesis except bacteria. Photosynthesis except photosynthetic bacteria is performed in normal visible spectrum. Normal visible spectrum. And that is, normal visible spectrum range is 390 nm to 760 nm, out of which plant uses or perform photosynthesis in between 400 to 700 nm. This is PAR, that is PAR, photosynthetically active radiation. Plant performs photosynthesis in between 400 to 700, 400 to 700. And if this light is passed from the prism, it divides into its component, that is VIBGYOR, violet, indigo, blue, green, yellow, orange, red, according to their increasing wavelength, according to increasing wavelength, V, I, B, G, Y, O, R. Light comprises of the 7 colors. This is increasing wavelength, increasing wavelength. If we consider monochromatic light, monochromatic, that is only one color. In that case, best light is red light. Photosynthesis, for photosynthesis, if single component is considered, then red light is best. Next is blue, that is optimum in blue, and the least in green. Least in green. Photosynthesis takes place least in green. Chlorophylls are green colored pigment. They reflect green light. They reflect green light. So if we arrange monochromatic light in order, then R, B and G. These are the 3 colors. Maximum, optimum and minimum. Maximum, optimum and minimum. If we study the absorption spectrum, as far as absorption spectrum concerned, blue light is best for absorption spectrum only. Absorption spectrum. In that case, blue is better than red. Blue is better than red. But as far as action spectrum concerned, in action spectrum actually output is seen, and in photosynthesis output is evolution of oxygen. So we see what amount the carbon dioxide is released by the plant. In that case, red light is better than blue. Red is better than blue as far as action spectrum concerned, and in case of absorption action spectrum concerned, blue is better than red. And absorption spectrum and action spectrum were studied by Mr. T.W. Engelmann. T.W. Engelmann. Below V, V, I, B, G, Y, O, R. Below V, ultraviolet light, UV rays. It is detrimental for the organisms. Above are infrared light. Above are infrared light, and useless for plant but useful for bacteria. Infrared light, that is above, are useful for bacteria but harmful or useless for the green plant. Green plants photosynthesis, again and again, it takes place only in visible spectrum, and it ranges between 400 to 700. That's why this is called PAR, that is photosynthetically active radiation. Now, duration of light. Duration, that is from sunrise to sunset, that is known as duration. Duration of light means from sunrise to sunset, that is photoperiod. It has no effect over photosynthesis. Photoperiod has no effect on rate, not photosynthesis. Light is available, till light is available, plant perform photosynthesis. If light is not available, photosynthesis will be stopped. But duration of light does not enhances the rate. Rate with respect to light can be increased or decreased by the intensity of light or quality of light, not by the duration of light. Duration of light, that is 12 to 18 hours light is sufficient. 12 to 18 hours light is sufficient for the optimal photosynthesis. 12 to 18 hours light. Carbon dioxide. In atmosphere, its range is 0.03 to 0.04% volume by volume. Volume by volume in atmosphere. It is roughly in this range. C3 plants fix 15 to 35 mg per decimeter square per hour. This is production of photosynthetic production with respect to the carbon dioxide. That is, plant fix this much amount of carbon dioxide, especially C3, and this is for the C4 plant. C4 plants are more efficient than C3 plant. Carbon dioxide, if we see this particular amount, it is with respect to plant is natural limiting factor. With respect to plant, it is natural limiting factor. It is natural limiting factor. That is, plants need more carbon dioxide for their photosynthesis. Though it is a greenhouse gas, this is the mistake of human being. That is, they actually involved in the deforestation activity. In deforestation activity, if the plants are not there, then who will fix the atmospheric carbon dioxide? If carbon dioxide in atmosphere, it absorbs solar radiation and responsible for the global warming. This is called greenhouse effect. This is known as greenhouse effect. So for that, human beings are responsible. But as far as plant concerned, if atmosphere has carbon dioxide, rich amount of carbon dioxide, it is beneficial for the plant, especially C3 plant. This is already mentioned. That is, C4 plants C4 plants show saturation at about 360, whereas C3 plants 450. C3 plants respond to increase up to this limit. Suppose if in a greenhouse, carbon dioxide is increased, plants flourish well. This is carbon dioxide fertilization effect. Applicable for C3 plants. their rate enhances quite dramatically. Their rate enhances quite dramatically. But again, if it is limiting factor or within limit, then only enhances rate. Now temperature, next is temperature. This is optimum temperature for the C3 and C4 plant. For C3 plant 10 to 25, for C4 35 to 40, 30 to 45. So with respect to temperature, directly proportional, that is rate of photosynthesis is directly proportional to temperature. But again, if it is limiting factor, then only. Water absorbed from the soil. Water is absorbed from the soil by the plant, and water is essential for all physiological process, not only for photosynthesis. It is required by all processes, especially enzyme controlled. So if water is not sufficient in plant body, overall living activities will be affected. Overall living activities or vital activities will be affected. But especially in photosynthesis, there is a direct role of water. It is involved in the photolysis of water in two ways. Due to photolysis of water, first of all, these protons are released. They are taken up by the carbon dioxide for the formation of CHO compound. When water splits or water undergoes lysis, it is a source of protons or hydrogen, and these hydrogens are gained by the carbon dioxide, then only any CHO compound can be formed. So first of all, source of proton and source of electron also. Water acts as electron donor in photosynthesis. We have studied the photosystems. When PS2, that is its reaction center P680, due to photoexcitation loses electron. This electron which is lost from the PS2 reaction center will not come back in this state, in this particular center. So in that case, water undergoes lysis and provide electron to PS2. So water also responsible for fulfilling the loss of electron of PS2 or photosystem 2. This is the role of water. And third, that is useful for us, that is, due to photolysis of water, atmosphere is enriched by oxygen. Oxygen is released from the water into the atmosphere during photosynthesis process. So source of oxygen, that is for the all, that is useful for all. Oxygen is useful for, beneficial for all. But for plant, water, hydrogen source, then only CHO are formed, and second, electron donor. Second, it is electron donor. Now come to the internal factors. In internal factors, first one is chlorophyll. Chlorophyll, light harvesting pigments. These are the main pigments, light harvesting pigment. Light harvesting pigments. Especially chlorophyll A. Because it is chief pigment of photosynthesis. Chlorophyll A is the chief pigment of photosynthesis. So if there is a deficiency of chlorophyll, definitely there is an effect over the photosynthesis process. But the content of chlorophyll is not directly involved in the rate, content of chlorophyll, the amount of chlorophyll. Still, the assimilation number is associated with the chlorophyll content. Assimilation number or photosynthetic number, that is amount of carbon dioxide in gram assimilated by 1 gram of chlorophyll per hour. The amount of carbon dioxide in gram assimilated by the 1 gram of chlorophyll per hour. Chlorophyll has no direct role in carbon assimilation. It helps in absorption of energy, absorption of solar energy. That is for this particular activity. So chlorophylls are the source of energy to the plant, but they receive energy from the sunlight only. And the last one, photosynthetic products, especially the Calvin cycle, is enzyme operated process, enzyme governed process. If end product accumulates, the rate declines. If end product accumulates, this is feedback inhibition. That is feedback inhibition. That's all for the photosynthesis. Now photosynthesis is over. Thank you.